and today we have very interesting paper by Joanne Wardell.
Yeah. Okay, yeah, so this is more like a kind of a neuroscience paper, but I feel like it probably impacts like the research that we do in our lab potentially. But this paper is looking at like blood volume change across different groups of neurons in the brain. So, just some background slides. So, there's this concept called neurovascular coupling, which is basically when different regions of the brain start, like different neurons start firing rapidly. Some kind of like fuel signal is released, and these things called microglia are kind of sensors waiting for these kind of events. And then when this fuel signal occurs, the endothelial cells widen and it allows like oxygenated blood to flow into that part of the brain. So I kind of wish maybe Jordan was here so he could explain this figure a bit better, but essentially that's kind of what happens. And so that
Because there are many questions there. There's blood-brain barrier. The blood doesn't actually get to the neurons. So how do the glia, like they take that electron or whatever, the oxygen, and they bring it to the like, but they go through the tunnels or like what like through pore like yeah definitely blood is insulated, it doesn't get
mm,
to the neurons, but
I
I'd like
think
to
there's
talk
some kind of channels or
like
like enzymes.
to know more about the mechanics of it.
I think it's still active research.
Yeah,
What
maybe.
people know about it.
Yeah, it's a fascinating process. It was really cool to kind of just see like different diagrams. This was kind of the most succinct like image I could find, but yeah, so essentially,
There.
yeah. Um, so yeah, and so neurons are firing, releasing these signaling molecules. So then these nearby blood vessels dilate and blood flow increases to that region, essentially through like a complicated process. And then so more oxygenated blood arrives in this area. And so it causes a change in the magnetic field in that area, and then the MRI machine picks it up. And then that results in a signal, which is called the bold signal, which stands for the blood oxygenated level dependent signal. So this is the kind of like the primary metric that's used in fMRI to map the activity in the brain.
You kind of skip this liquid, but it's basically when the oxygen is attached to hemoglobin doesn't have the magnetic amount, but when it releases the oxygen, oh, suddenly it becomes detectable, and that's what we detect.
Yes.
That release of the oxygen.
Specifically, they all align in the same direction, and that affects the field.
Oh, yeah.
There's physics
And then they fall on.
that
Sorry. And then, so a lot of the times, people are using this thing called a hemodynamic response function, which is kind of a mathematical model of this process of oxygenated blood arriving and leaving the area of neural activity. And so people will take this function and then convolve it in a way with their um, bold signal. So like I said, in the beginning, there's like a stimulus, and then there's kind of how this function is characterized. There's like an initial dip in the bold signal, which people characterize with like usage of oxygen, and then it peaks, and then there's like a post-stimulus undershoot. But this is kind of what this function looks like that people use in their analyses. And so what people will do here is like when they have a task-based study, they want to understand how different voxels are responding to the task. Is the activity or change in this voxel positive or negatively correlated with the task that's being studied? So they'll come up with something called a task vector, which is kind of like this binary ones and zeros vector that maps to the timing of the task that is being performed in the MRI or any study.
Can you explain more? I don't know if you put like wording are confusing. What do you mean by task and what? Like, yeah, just a little bit.
So maybe someone's sitting in the scanner and they want to understand maybe like how someone is tracking or responding to a button press. So for the parts of the study where they're not pressing the button, it'll be 0. And then for the parts of the study where they're pressing the button, it will be 1. And so we use this task vector to analyze the voxels during the task. So the vector is on during the task, meaning 1, and then the vector is off, meaning 0, when the task is not happening. And so there's this href function here, and this is obviously a continuous function, so we have to evaluate it at the sampling rate of the scanner. So once you evaluate it to get the same number of time points as you did in the task, then people will convolve this with the task vector. And once you have this, you have something which is referred to as a predicted bold value. And then you have the actual bold value for a particular voxel. And what you can do is take these two values as a coordinate and plot those. And then you can try to fit a line to it using a linear regression. And then when you estimate the beta weights for that line, the slope of that line represents the magnitude of that particular voxel in response to the task. So what you get is the input is like a 4-dimensional fMRI scan, and then the output is this 3-dimensional task, kind of like a task mapping of the brain at each voxel, how it responded to the task. So it's kind of a lot, but it's used in this paper, so yes. And then another thing is this concept called functional connectivity. So people will take these 4-dimensional fMRI recordings and then they'll do ICA to get different components. And each component, after filtering and processing, we'll consider as a brain region. And we can find the bold signal of this region over time by just averaging the activity across all the voxels in the region. And then we can correlate those time series to get something called a functional connectivity matrix. This is just like an example.
So I do want to point out something that Vince always corrects me on. This is functional network connectivity. It's different. Functional connectivity is widely discussed in literature, but it's different, right? You have either an ROI from an atlas where you take the raw values from the voxels and aggregate them in the ROI and then use that for correlation, or you do voxelized correlation, which is quite popular for functional
like
connectivity.
seed-based.
Seed-based functional connectivity as well. They're different fundamentally from ICA because we have spatially continuous intrinsic connectivity networks. This is like one of the formal names of them. That's a slight difference. Vince prefers it. So it's something that he corrected me on for years. So it still corrects me on.
Okay.
Well, because those are functional networks.
Yeah, they are.
Okay.
ICA. And they already did some conclusion.
So functional connectivity is computed on voxels and so
Voxels
There
are
are many
ROIs.
ways. Yeah, there are many ways.
Yeah, it's just you should separate it from the ICA-based term. It's because it is a fundamentally different thing. Voxelized functional connectivity is popular. Seed-based, it's quite a popular ROI level.
So I can see something specific to ICA personalization.
Or to other things that derive functional.
The way to put a space in the FNC. Functional network is one word, and then space connectivity. It's the connectivity of functional networks.
Yeah,
that's what.
So you might see in some of the ICA papers they call them intrinsic connectivity networks. That's where that comes from, right? It's this idea that ICA is detecting groups of voxels that are
those are functional networks.
Yeah,
and this is connectivity between those networks. It's this nested use of the word network, like me coming from computer science.
Yeah, it's a mess. It's like the terminology is annoying. Yeah,
so but we do that. We do that in language.
We do. But that's just a subtle thing that I did not get wrong for years.
Yes, thank you. Okay, so yes, this is the functional network connectivity matrix. For example, like when you get the regions from whatever analysis you're doing, and then you compute the correlation between their time courses, you can get this kind of matrix. So I think historically people consider positive correlation between these regions as representing some kind of shared functional network activity. And whereas negative correlation is associated with some kind of anti-correlation between regions, representing like competing cognitive states or processing, if it's like dynamic efficacy. So another thing that's another technology that's used in this paper is called neuropixels, and it's this type of shank that's actually embedded into the brain of a live organism, and it's pretty small, and it directly measures hundreds of or thousands of neurons in a moment, and it's a direct measure of the electrical activity from the neurons. And they have an ultra fast resolution that captures the neuron firing within like milliseconds timing. Oh. And then, so we in our lab use like fMRI or like EEG or maybe MEG, but in this paper they're looking at something called functional ultrasound imaging. So it's typical ultrasound, which is used for different scans. But it's essentially an imaging technique that's based on the physics of sound waves to track moving blood cells in the brain. So it uses the Doppler effect to calculate frequency shifts in moving blood cells to build a real-time map of the cerebral blood volume. So from this modality, we're getting the blood volume time series.
Does it actually see individual cells, like individual hemoglobin cells?
So from what I understand, I'm obviously not like a physicist, but
Well, from the
it shoots like it shoots like a sound wave at the blood cells, and then the echo that it receives, the difference between the timing is how they calculate the movement of the blood flow.
Little tiny radar.
Yeah, and then
like, what's the resolution? Does it actually based on cell level?
It's more likely to detect the flow of the individual.
yeah, it's used to compute the volume, so it's not really looking at cells. I'm not sure though the resolution.
Yeah, okay. That makes sense.
Okay, and one more kind of concept is this thing called whisking in mice. So mice have these little whiskers on the front of their face, and then they move them back and forth depending on like if they're in an aroused state or like if they're becoming alert. So you can kind of compare this to like eye dilation in humans. A mouse can map their environment through these whisking movements. They can detect like sounds, vibrations, etc. So again, like when they become alert or maybe curious, the brain releases these neuromodulators, which triggers this whisking behavior. So they're going to use this in the paper to kind of track arousal globally in the brain and kind of see how it corresponds to other measurements that they're taking. So from what I picked up in this paper, they did 5 main experiments. One is where they're tracking kind of this arousal of the brain globally with this whisking movement as they observe the mice. And then they go about classifying the different neurons that they pick up from the neuropixels as being arousal positive or arousal negative. And they also they build this model that encapsulates the differences between positive and negative cells and reach some conclusions there. And then they analyze this model in their 3rd experiment when the mice are asleep and awake. And then they kind of look at the overall global distribution of these positive and negative neurons. And they use this to predict, they also look at the regional blood volume. So in their first experiment, they looked directly at the this I don't know how to pronounce this, if it's like FUSI or FUSI. I guess I'll say FUSI. Okay. So yes, they looked at the FUSI recordings of these mice. So this is a full brain imaging modality. So they can like parcellate it out to different regions of the brain, or they can kind of I guess look at the regional volume. And then they also, at the same time, are taking the neuropixel recordings from 2 different regions of the brain. And they're also picking up the whisking during these experiments. I'm not exactly sure what that data looks like, but there's some kind of whisking time series. So there's the blood volume time series, there's the neural activity time series, and the whisking signal over time. And the mice in this first experiment weren't given any stimulus, and they monitored the blood volume across all the regions, and they were paying special attention to when the mice just started randomly whisking. So when they did that, I think in the first experiment, they were just kind of looking at the plots, especially just around these whisking points to see what was the global activation of the brain as well as the volume. And then in the second experiment, they went about labeling the neurons that they recorded from NeuroPixel as either arousal positive or arousal negative. So how they did that is they took the NeuroPixel time series for each, and for each neuron that they recorded, and they correlated it with the whisking time series. And then if that was a positive correlation value, it was labeled as a positive neuron. And if it was negative correlation, it was labeled as a negative neuron. And from there, they basically from each neuron group, either positive or negative, they averaged all those neurons time series into one. So they only have one positive time series and one negative time series. And then from there, they regress each one of these time series against this Fusi time series to get these HRF weights. So they're not using the canonical HRF function, they're learning one for both the positive and the negative. So they take this set of HRF weights, which they learned from this Fusi time series, and they convolve it with each one of the arousal time series that they found from the positive and negatives. And from there, that gave us like a predicted Fusi time series for the positive and negative. And then they correlated this estimated time series of the blood volume with the true to get a correlation value. And they repeated this experiment in a scenario where the mice were allowed to like drift in and out of sleep. And so they they wanted to look at the positive and negative cells in the sleep state versus the awake state. And they used the labels that they learned in the previous experiment to label these neurons. They didn't have to redo that step. And they also just used the HRF weights that they learned from the previous experiment. They convolved these with the positive and negative labeled activity from the NeuroPixel. And then once they got that predicted, they correlated it again with the original to get like a predicted value. So there's another data set that they have for the NeuroPixel measurements, and I think they might have found this, or maybe someone else did, I'm not sure. But it is a NeuroPixel data set from 55 different regions in the brain. And so this allowed them to do kind of a global comparison of actual neuron activity across the brain. So so for each region, they looked at the correlation of the activity with the whisking for each region and each neuron in each region. And then they applied the labeling in the similar fashion that they did in the beginning, but they were able to do this across the whole brain instead of just 2 regions. And so once they had the labels for the entire 55 regions for either arousal positive or arousal negative, they took the ratio of positive versus negative for each region, and then they mapped these proportions across the whole brain. Now in this one, they did one more experiment, which they correlated so they looked at the ratio between arousal positive and arousal negative cells. And they correlated this to the magnitude of blood volume change during the Fusi recording of that region. And they did this across all regions. And then they compared this to the bulk firing rate time series, which is basically all the entire neurons time series averaged together. They correlated this time series with the original Fusi recording for that region. Okay, so this is what they found. So as they observed the Fusi recordings during the whisking task, they noted, or not a task, but it just occurred when they noticed the whisking was occurring. They noticed that the blood volume across the entire brain increased at once. So as you can see in this image on the bottom in panels F, G, and H, there's like this straight red line that's all the way down on all three of those. So that's kind of showing the global increase in neuron firing during the whisking activity in the mice. So they just observed this overall positive firing activity across the brain. So here they correlated the whisking with the Fusi time series to label the neurons as arousal or not. And then they learned the HRF function for each group and then convolved it with the average neuropixel time series for each group, either positive or negative. And then they got the predicted volume time series from that, which they correlated with the original one to get a correlation value. So they found that the two time series that they did this in were correlated for the positive and negative. But they also did this experiment for the bulk firing. So in the bulk firing, they show that if they simply average the activity in the neuropixel across all the neurons without considering the plus and minus, and then convolving it with a separate HRF that they learned for the bulk firing rate, and they correlate that with the original Fusi recording, that they're not finding a strong correlation. So you can kind of see that here in the G. And then compared to figure 3, where they do this entire process again, where they learn a different set of HRF weights for the positive and negative arousal neuron groups, and then they convolve these with the HRF and then compare them with the actual Fusi recording. So here they're separating the signals into the positive and negative neuron groups. And then here they're actually, after the convolution, showing that the predicted time series is a lot more similar to the original than maybe this bulk prediction. And this is kind of an aggregate display of that activity as well. So they were finding that their separate modeling, in which they consider the positive and negative groups of neurons that fire globally with arousal, have a higher correlation value than when they just do this bulk experiment.
Sorry, John,
Mhm.
can you explain again what these groups of neurons, arousal, positive, reacts in a different way than negative? Also,
Yes. So the arousal plus group is the neuropixels that were positively correlated with the whisking time series. They receive a positive label. Yes. Any other questions?
it means that positive neurons have shorter HRF.
They have a different HRF. They learned a different set of HRF weights for the positive versus the negative. So they each, yeah, they,
I see, yeah, okay.
The negative were just the remaining neurons, or
no, the negative ones are, so the correlation can either be positive or negative
or zero?
or zero. I'm not sure what they do with the zero.
That's what I was getting at. I don't know if
Um,
there's anything that's zero correlated, but
yeah,
the boundary around zero would be interesting.
that's a good question. Yes, so, yeah. Any other questions? So Here they repeated the experiment with mice going in and out of sleep freely. They used the labels from the previous experiment to assign the arousal positive and negative, and they averaged for each group and then convolved these with the learned HRF weights from the previous experiment. Then they correlated these predicted FUSI recordings for that region with the actual FUSI recording, and then they did this separated into the sleep and wake states. So here they're looking at the FUSI for sleep in a region and then compared with the predicted for the positive sleep for that region. So for example, in region 1, they're looking at the predicted positive sleep versus the true FUSI for that region. And then they're looking at the predicted negative sleep versus the true FUSI for that region. So as you can see, on this figure, they have separated the neurons by arousal plus and minus. You can see the overlay at the bottom of section B for blood volume.
You point there.
Yes, yes. Yeah. So for blood volume and the predicted, they also contrasted this with the bulk prediction where they do the same thing, but without labeling as plus or minus. So basically when they did this for the sleep task, their combined prediction was better than the bulk prediction, but not significantly better. Okay, so here they're looking at the ratio of positive versus negative neurons throughout the brain in different regions. So for each region, which they parcellated using some atlas, they counted the number of positive versus negative neurons in that region, and then they took their ratio. And what they found is that these regions are present all over the brain. And here they're just kind of plotting like where these different regions fall with respect to how many positive neurons that they have and how many of these negative arousal neurons that they have.
None with zero, though, looks like, as far as I can tell.
Yeah.
So there's always some proportion, is their argument.
Yeah. So in panel D, they aligned the time with the whisking onsets and they plotted the average firing across the arousal plus in the region. And then in panel E, they aligned the time with the whisking onsets and they plotted the firing rates across the arousal minus in that region. So arousal minus during the whisker arousal, it activates in a negative way. During the arousal, they decrease towards zero in response to the whisking. So even though it's reacting such that the firing is trending towards zero or even below zero, it's still a response to the whisking. And I think it's synchronous with the increase in firing with the positive, even though they're in different signs.
Right, and something you said is confusing me still. So you said negatively activating, but the description of these is that it's this pixel time course is negatively correlated, right?
Yeah, I guess that would be
That's more what it is, right? So when that, when the thing that you're correlating with goes up, the associating thing is going down,
okay.
Right? So okay.
So in the last experiment, they are comparing the bulk model with their combined prediction model. And in the panel C, you can see typically the error for the bulk prediction model is usually higher than the error for the combined prediction model. And so how I think that they're doing, how they're displaying that in panel D is that, so I think C is the raw count of all of the positive and negative neurons. But then D, they normalize by the number of neurons in the region. And if something is higher or more positive, that means that there was more positive arousal plus neurons. So the population ratio explains the regional differences in blood volume response, which they found was better than the bulk firing rate. Yeah, so there's another claim that they're kind of, I think, implying is that these the differences that they find in these regions aren't necessarily because they work differently at the neurovascular coupling level, but it's because of the differences in the amount of these arousal plus versus arousal minus neurons. So I think the takeaway and the one conclusion here is I think previously if researchers saw a negative correlation between two brain regions, then they assumed that these two regions were hardwired into competing cognitive networks. I mean, and this paper suggests that a negative correlation can appear simply because one region has a higher abundance of these arousal minus neurons and that the other one has more arousal plus neurons. So maybe the network behavior that we see in human resting state fMRI might actually just be a global map of our shifting arousal states. And if we see two positively correlated regions, are they actually tracking global arousal together, or are they working together in a shared functional task? So I think this paper kind of
that's very intriguing argument. Is that something that they conclude in the paper, that this can translate to human fMRI?
This I don't know if it translates to human fMRI. This is a very new paper. Yes.
Okay.
Oh,
no, it's very interesting. Well, we could talk about it.
I think there could be implications
Yeah.
if it shows to be true in humans, but it's a very new paper. I don't know if it's been replicated anywhere else. So I guess the takeaway is when we work with fMRI, every voxel or ROI is a mixture of these like arousal, positive and negative contributions, which we obviously can't separate directly because of the temporal and spatial resolution. But so maybe in future experiments, we could mitigate these effects by having some kind of like continual arousal proxy, like where we're tracking eye pupil dilation or other measurements of this in humans, and then we could regress that out. I don't know if this is related to like local signal regression or what. Maybe it's why people do things like this, or and then maybe in modalities that are actually capturing direct neural activity like EEG and MEG, they could be aggregating these positive and negative arousal neurons in some way that's reflected in further analysis. I'm not sure, but maybe. Yeah, that's it.
Very cool. Definitely like very intriguing and suggestive for sure. Yeah, I mean, certainly I think the evidence in mice is compelling. Yeah, TJ's going to say something similar to what I'm going to say, I think.
Do you want to just say it, TJ? I don't know if you have a microphone.
Yeah, sorry everyone, I don't have video today. Yeah, no, Brad, go ahead. You go first.
No, you say
it. No. It's just
No.
very interesting that the results come back and saying it's a mixture of signals, just because we assume that it's a mixture of signals anyway. We have a bad measuring stick. And so I don't know necessarily how that impacts like multimodal like analysis. I don't know, Brad.
I was going to say almost exactly the same. That fMRI is the resolution is very, very different, right, from this. So you're looking at larger populations of neurons.
Mm-hmm.
It's still very interesting, right? Like if these populations are dominated by these negative arousal neurons, like there could be an argument for this like intrinsically negatively correlated signal. But it sort of remains to be seen, right? I think it's not a human versus mouse story more than in my mind, a resolution, what these things are actually measuring story. We do, and I also, one thing about this paper that I at least didn't see in your presentation was, do they break these up into different frequency bands, and are they looking at, because we do filter for heart rate and changes and that kind of thing, like, so some of these arousal signals will arrive at different frequency bands, right?
Mhm.
And
I was just looking at the resolution online, and it says that the temporal resolution for the SPUSY stuff is on the order of 1 to 10 hertz. So 1 to 10, 100 milliseconds per frame, which is faster than fMRI.
yeah, what's the spatial resolution is what I was referencing.
It's 100 to 300 micrometers, so
Okay, much smaller.
much
than-
smaller.
Yeah, voxels are 3 millimeters, right?
Mhm.
You know, sometimes.
Well, I mean, 0.7 right now.
Yeah, the best. Yeah. So it's an interesting story, but the
It's
scales are
1,000
so
times
different.
smaller.
Like I wonder how, I think there's remains to be said, like how it will translate from that very small scale to
Yeah.
that very large scale. But it's very compelling. It does beg the question, right?
Yeah.
But I don't understand like the negative part. What my understanding of fMRI, what it measures is energy intake, and there is no negative. Neurons do not produce energy. It's
Well,
like no matter how you correlate, you still consume. You need energy. You
Right.
need to take that.
Just that the bold is like decreasing during that period, right? So it's the blood, and I imagine
So
it's
taking
similar.
a lot of energy.
We're taking, or the slope is like decreasing, right? Compared to like an increasing one. It depends, like, because when you're looking at correlation, like you need to look at the direction of each signal, right? Because
Oh, okay. So you stop taking energy. Like the neurons are not active there.
or it just means that the coupling of the slopes is opposite, right? So it could be that in one region you're seeing a decrease, but that means in the other region you're seeing an increase, and vice versa, right?
Sort of like if the brain was like all the blood was contained, and you imagine there's no more blood added, and it's like moving around. It's like either moving towards or away.
Well, but that's practically what's going on.
Yeah, as well. Yeah. So
But then there is
they're
more blood entering though. So then it
And it's in the bare body.
yeah, certainly it's very interesting, and I think multimodal, will maybe answer some of those questions for us in some degree. I do think, this doesn't make me worried about that we're doing something fundamentally wrong in fMRI. I guess is the answer. Like it's possible we are, but more evidence is needed.
So wait. I slept through it or something. I was looking at the slides, too much text. Not a critic, but just. So what is the conclusion that we can draw from that? That the bold signal does not represent actual blood flow, or
I don't think that's the conclusion.
I think from what I can tell, it's some observations that they have at like the neuron level, but I'm not sure how it applies to the network level.
Were you saying that fMRI could also be seen just as like levels of arousal, right? It's like how much. Will you explain that? What did you mean by the fMRI and the arousal state?
Which one? On which slide?
Your last ones, your takeaways. Or
Okay.
maybe you just said it out loud.
Oh yeah. I mean, so fMRI is like an abstracted view of the brain, which comprises neurons that are potentially doing this arousal plus and minus. So we're observing that somehow in aggregate, but how to actually separate that in fMRI is unknown. And I'm not sure what to conclude from that, but
Right. So their conclusion is that on the neuron level, there are groups of neurons within these like well-defined ROIs that will be anti-correlated or correlated with this arousal signal.
Yes.
Right? So it's really like the interpretation for rest of fMRI also needs to be seen, right? But for arousal specifically as a compound, it does potentially bring in some questions. So TJ said that there's a follow-up paper, Taylor Bolt, about arousal regression. But
Yeah, hey, there's the camera. Sorry for that. Yeah, there was a paper where they actually looked at arousal, heart rate, I believe movement, MRI, and sleep maybe, but it was like super multimodal, and they were trying to suss out more of the global signal and what is captured within it. And yeah, I think it's more new and not as disseminated as well. So I'll post it in the chat.
Yeah. think it's super interesting. Like I think the arousal compound you could be investigated in fMRI without even thinking about this like neuron level story, right? Because it certainly could be a compound.
So there is neural activity, there's blood flow, and what is arousal in this context?
Whatever human equivalent of whisker sensitivity. So you talked about eye dilating, heart rate increase.
Yeah.
Uh,
And it's not like it was
stress
triggered.
related.
But it's all
occurs.
driven by neural activity
Yeah.
anyway.
Right.
And then a feedback loop, of course, blah blah blah. But it's just mechanically, I don't see anything present like a vital force or anything like that. But neural activity and blood flow. Like, how do we combine those two to get the arousal explanation?
Well, I think the story was more that arousal specifically shows this like split between anti-correlated and correlated populations of neurons, right? And so if you're looking at measuring arousal, just doing them all in bulk is the wrong way to do it. That's, I think, the conclusion.
But they were able to use that arousal mapping to accurately predict the blood volume in this scan of, yeah. So it was a predictive feature.
Well, it would be more similar to like a task, I think, like where someone is doing like a thing in a mouse, so they had this arousal that task that the mice.
But in a way, it's intrinsic because the mice are not being stimulated, but they go through periods of being aroused for whatever reason. Same, maybe humans as well, by their own thought.
You have a stressful thought, I dilate, start speeding, you think about your ex, and oh my goodness, yeah, that kind of stuff.
That's how I wake up.
Won't tell.
Otherwise, there's no way to wake up.
Yeah. Oh, you wake up stressed. I think. Yeah. Yeah, I think there is a question, right? Is this like a problem for fMRI if the rest of fMRI were standing that sometimes we are getting these the person has a depressive thought and their brain changes, or they have a stressed thought and their brain changes. So that's just part of the rest signal, I think.
I was thinking something completely unrelated and related. Just trying to connect with the postdoc's name who gave a talk about Fourier, the waves in the brain.
Oh yeah,
right?
yes, Joe.
Yeah, and I was thinking about the waves in the brain. If we think about the physics, are very different from the sound waves and things like that. In a sense, they're more they're like fire waves, or you have dry grass, you fire it because the neurons they used up the energy. They need time to get excited or to grow back the grass for the next wave to come. Versus like, oh, the wave just went through.
I think that's a good
if it went through, it used up. It's a different kind of wave, so it doesn't line. Like, I mean, it's still a wave, but blah blah blah.
Yeah.
An abstract notion. We can decompose anything in Fourier. It have to be a wave. So there's trickery there. But here, so I'm thinking like if neurons have used up their charge, the action potential, so they fire, they need time to recharge and fire again. And
yeah, it could be as if the temporal resolution on this increases anymore. I mean, you're kind of getting beyond what the vasculature could do at a certain point, right? But,
yeah, exactly. It's a different. But then I think the governor who decides where to pour blood acts not on per neuron basis, probably. It's a symbolic signal.
Yeah.
Okay, like, I don't think neurons work alone. Maybe there's some raw neuron that's just doing
the one that was So I just have one in my brain.
Yeah. Like, why you all guys? I will be doing.
Yeah, whenever I get a weird idea, I send you 20 messages at midnight. That's not fun.
No. Keep doing
Yeah.
I'm curious to
Yeah.
see this in sleep. Like I know we can be slightly aroused in sleep, but not as aroused as like waking. So, but they also.
Oh no, we're very aroused, and it's just that's why you have the coupling mechanism that turns off the signals from your brain to your body. Otherwise, people who have a problem with that, they can kill a person nearby them by accident or something.
Right.
Or like all the movements and things like that. So the brain is highly, highly, highly active. Yeah, it's maybe more.
That's a little scary. Yeah.
Just enjoy the movies when they get into your conscious. They manage to get through.
I have a question about the fMRI again. So if we are using not the fMRI, F and C.
Oh yeah.
So if we're using an ROI atlas that has functional networks, like I think Schaefer has them, we also can.
No, but this is an arbitrary carving. And yes, TJ, now you can participate in this semantic argument as all philosophers like to do. But we take the brain and arbitrarily carve it in voxel spurs. Like millions of neurons, this will be a group. Arbitrarily, maybe we would have shifted it by half of the voxel, the entire grid, and it would have been better for this voxel, but not for that voxel. So that's first arbitrariness. But then you're saying, oh, we will group those networks on voxels arbitrarily into an ROI based on
They do call it functional networks though, which is
yeah.
But I think we also do an ICA on this voxels.
But in ICA, we're deciding yes, voxels are done. It's already irreversible damage. Yeah, we can't do anything. Yeah, but at least there are two ways to like arbitrarily carve them or versus the correlational or data.
Yeah, so ICA, the spatial maps, if you unthreshold them, they're fully continuous right over the brain, right? So every voxel contributes, but some voxels just have basically zero contribution, right? That's where we, if you generate your spatial map figures, you choose a good threshold. Otherwise, you just get all these speckling throughout the brain, and in fact, you'll get the entire back.
But this is a display issue.
It is
when we look at the time course, we assume the entire brain of that component acts together.
Right. But even the the map itself is like a distribution. Is how I think of it.
You had this figure with our IC components somewhere.
Oh yes.
Oh yeah.
In the beginning.
Yeah, that's heavily thresholded.
Heavily thresholded for display only. This is just display.
Yeah.
Each color it's an entire like the time course, as Brett said, belongs to the entire brain.
Yeah.
I don't know.
So that's the distinction with ROIs. I think Webheads likes to call it functional network connectivity versus functional connectivity. Even the functional networks that are from an atlas, we would probably call functional connectivity. It's weird.
Yeah, because the true functional connectome would include every single neuron in the brain, right? And that's why people are trying to estimate the connectome with the ants and stuff like that. Yeah.
Go further. What do you mean?
I think let's talk about the gut. Let's talk about our spinal cord. Let's talk about breathing.
Oh yeah,
the functional connectome in those regions.
I mean, we study the brain. Why do we only have to stop that?
I agree. Yeah, there was a really cool.
So I don't know. I would just think that we silo ourselves already. Why are we siloing ourselves only to the brain? Even I probably have bad internet connection. I apologize.
Yeah, you're definitely cutting in and out. we heard the end of that though.
Sorry about that.
No, you're good, but you're totally right. I think there was a recent paper looking at the brain spine connectome, which I think looking at these other neuron populations will be really interesting. I was really interested in the gut neuron population for a while.
You worked with Jordan on that, right?
We worked on the microbiome, but yeah, I'm also separately interested in the neurons in the gut.
More so, why are only studying the brain? Why does the connectome stop at neurons?
Yeah, and why does this positive negative story stop at the brain? I think it would be interesting to see it in the spinal cord potentially in a heavily neuronized regions. Neuronized, just better nervous. I don't know.
They're in the network.
Yeah. But the gut has a huge population. Of course, around the spine. Hands, fingers, your eyes.
Aren't they just the axons, like dendrites, not actual ganglia? Everything goes in and out. Not.
Yeah, that I know.
We don't have any processing in our fingers.
There's no processing of the fingers. But
it's mostly. I thought it's just long wires to get the signal back and forth. and that's why there is the delay, because what is it, 1 meter per second or 2 meters per second, the actual speed, and so you can burn actually before you realize that.
TJ, correct me if I'm wrong, but are there like actual neuron like axons in the gut, or full neuron like cells in the gut, like not just I remember reading that somewhere. Yeah, I don't know.
Yeah, are there ganglia there that actually,
Or is it all white matter?
Yeah.
Essentially.
connections? We can't hear you.
I don't know. I'm gonna have to look that up. Yeah. I know there's a whole microbiome. Sorry.
Yeah.
Okay, well, thanks,
Yeah,
Joanne.
thank you.
Thank
Very
everyone for
compelling and
participating,
interesting paper, yeah.
for your thoughtful questions today. And there's an interesting talk right now at DIMA next.
I think it's online.
Oh, it is online.
Yeah.
Oh, okay. I was. The abstract looked interesting. So.
Yeah, if Armin posted it, it's mostly online, I would expect.
Oh, it is about 7 Tesla high high magnetic field MRI. And, yeah, what they did with my work at MRI here. So, yeah. But okay, thank you everyone who was with us.